Good afternoon. I'd like to welcome everyone today to our inaugural celebration for 4 of our named professors. As many of you know, we moved to a new format this year where we are honoring a number of chairholders in each of the ceremonies. And instead of people each giving a scholarly talk, they've been asked to talk about important events in their lives that may have influenced how they came to study what they study and who they are today. Fred asked me whether it was okay if his comments were at all humorous. I said, of course, humor was allowed. But it's been interesting to hear the speeches at the 3 other investitures we've had in this format, actually preceding 4 investitures, I guess, held this year. I've learned how racism, religion, mentors, collegiality, catastrophic events, family dynamics, and Northwestern's institutional breadth have shaped our professors' lives. It's really been a rewarding discovery to hear these heartfelt accounts of people's growth as scholars. So I'm looking forward to our talks today.
Jonathan Widom was appointed as the William Deering Chair in Biological Sciences in 2006. Can you begin to see why we've gone to this new format where we can honor multiple people at once? We were a little behind in actually formally recognizing our chairholders. So 5 years later, we are here to honor him with this chair. I began to see as I'm doing this introduction, there's a little bit of a theme. Not with Fred, but with the other 3 of you in Champaign-Urbana. I didn't actually know John at Illinois, but I knew of him because he collaborated with people that I did know. He was at University of Illinois, Urbana-Champaign before he came here. The chair he holds is named for the founder of the Deering Farm Implement Company, and who was the chair of the Northwestern Board of Trustees. Deering was exceptionally generous to the university. It's funny to describe him when you go to you can look up these various things in the archives. He was exceptionally generous donor, giving more than $1.2 million during his lifetime, which of course by today's standards we think, really? And that built a library and did all this stuff? But that was a lot of money then. And they gave very generously to the university, including probably the most notable thing that holds the Deering name, other than John's chair, of course, is the Deering Library, which is a very visible symbol of their philanthropy. The previous holder of the Deering chair as a permanent appointment was Nina Schwartz. It is my great pleasure to be here to recognize Jonathan Widom as the William Deering Chair in Biological Sciences. I would now like to invite Polly Mayo back up to introduce John.
It's my great pleasure to introduce John Widom to you. And as you've just heard from Sarah, there are actually many interesting, I think, commonalities and connections between this and my last introduction, including Cornell, chemistry, chromatin, Urbana-Champaign, and moving to Northwestern. So we seem to have a theme developing. John received actually his undergraduate education in chemistry at Cornell before pursuing his PhD in the Department of Biochemistry at Stanford University under the direction of Professor Robert Baldwin. And it was there that John began studies on the bending, the condensation, and the packaging of DNA molecules. His subsequent postdoctoral studies were performed in the laboratory of the Nobel laureate, Sir Aaron Klug, at Cambridge University in the Laboratory of Molecular Biology, where he studied the structure of chromatin fibers that were from vertebrate cells. John then did join the Department of Chemistry at the University of Illinois in Urbana-Champaign as an assistant professor, where he also held an appointment within the Beckman Institute. And we were really most fortunate, again, to entice John to move to Northwestern, to the then Department of Biochemistry, Molecular and Cell Biology. And it's hard to believe that that's almost 20 years ago now. John holds a second appointment in the chemistry department, and he's taken many leadership positions in his time here within the university. He served for 6 years as the chair of BMBCB. He directed our biophysics training program for a number of years, directed our Center for Structural Biology, and that theme continues as he currently directs the Keck Biophysics Facility, as well as the Physical Sciences Oncology Center, one of the very new and important centers here at Northwestern. John's research efforts are aimed at developing a defined mechanistic understanding of gene expression within cells. And much of that work has involved trying to understand how nucleosomes, proteins that package the DNA, influence gene expression within cells. John's lab performed some of the very vital early work that showed these structures that were thought to effectively condense DNA and make it inaccessible were in fact very dynamic structures that provided for flexibility in gene expression by providing for accessibility to regulatory factors. In later work, John went on to ask questions about how it is that nucleosomes become positioned where they are within the DNA of our genomes. And he made the really, I think, surprising and certainly important discovery that much of the information for positioning nucleosomes resides within the sequence of the DNA molecules of the genome itself. So he's gone on to really do, I think, seminal work on trying to understand what are the rules that govern where nucleosomes are positioned and what is the physiologic importance of that positioning for gene regulation. And in fact, John also continues to work in the area of DNA conformation, DNA mechanics, an area where he really started his research career. John's work has been recognized by many awards and honors, including the Cyril Scholars Award, the Presidential Young Investigator Award, and many, many named lectureships, most notably the very prestigious Van Leeuwenhoek Lecture at the University of Leiden. John publishes in the very top journals in the biological sciences and has held visiting professorships at both the Rockefeller University as well as the University of Rome. John has taught courses from the very beginning undergraduate level to the advanced graduate level at Northwestern, mostly in the area of biochemistry and biophysics, and he's currently teaching courses in molecular machines and chemical biology, and in fact told me that he's lectured in both of those courses earlier today, so this is his third lecture. John is an internationally recognized leader in the field of gene regulation, and I suspect he has the frequent flyer miles to prove it. John has made truly seminal contributions to our understanding of nucleosome positioning and nucleosome dynamics. And as well, John is really an incredibly thoughtful, insightful, helping and giving colleague at all levels who is always willing to contribute to the greater good, I think, of both the department, the college, and the university. So John is truly a very deserving of this important recognition for his outstanding scholarship. So I'm very delighted to be able to introduce John Widom as the new William Deering Chair in the Biological Sciences. John, congratulations, and please come forward.
Thanks so very much for the kind words. I'm humbled and somewhat embarrassed to be presenting my little story today. I'm humbled because I'm well aware that the previous holder of this chair, Nina Schwartz, who we heard mentioned, was a true pioneer in science and a giant in her field, setting a standard that I can only aspire to. I'm a little embarrassed, in part because I'm not accustomed to talking about myself, and in part because my story is so completely ordinary. There's just nothing remarkable about it at all. It's dictated in no small part by who I was born to and where I grew up. And I'm only going to show one map. And what I want you to see about my map is that Ithaca, New York is not on the map. In contrast to big cities like Sturgeon Bay, Wisconsin, for example, Ithaca, New York is not on the map. And so basically it's a little place with nothing noteworthy except for one thing, which is Cornell University. And what this meant as a practical matter, here, maybe I can turn the map off. What this meant as a practical matter was that growing up, everybody I knew was an academic. My parents are academics, their friends were academics. The people that I knew, the kids that I knew were children of academics. And so literally I had no idea at all about other careers in life. And so basically I was born onto an escalator leading to an academic career, and all I needed to do was not fall off. And I managed to do that to some extent, and here I am. So I've had really good fortune in my choice of parents and in my choice of where I grew up. But several other people, too, have especially influenced my career path, and I would like to acknowledge them here. Early on, while I worked in another lab at Cornell, I spent time with Elliot Elson and his group. This is while I was an undergraduate. And at that time, Elliot Elson, together with Watt Webb, had just invented FRAP experiments and fluorescence correlation spectroscopy, literally 15 and 30 years ahead of their time, respectively. And they were using these experiments to make novel discoveries in cell biology and biophysics. And I was, as you might imagine, impressed by the really imaginative nature of their work, and especially by how on almost any scientific topic, Elliot in particular seemed to know what were the key discoveries, what were the key unanswered questions, and what were the problems in thinking about and making progress on these problems. And then in addition to being a really great and original scientist, Elliot was also highly cultured and incredibly nice. And so, of course, I wanted to be like that too. And so I learned from Elliot that he gave a lot of credit to his PhD training, which he had received with Buzz Baldwin at Stanford. And so I applied to that department, and by a great miracle, I was accepted into their little graduate program, and I did in the end join Buzz's group. Now, in addition to Elliot Elson, Harold Chiraga and Gordon Hamas, and also Stuart Edelstein, in whose lab I worked, were highly influential to me. Each of them helped shape my choices, both with the broad field of biophysical chemistry and also about where to go for my PhD and with whom to work. And they all said that I should go to Stanford and work with Buzz. So looking back, I can tell you that it's not that easy to become like Elliot Elson. That's too bad. But in any case, going to Stanford's biochemistry department and signing up with Buzz was a fantastic move. After meeting him the first time, it was obvious that Elliot was right, that Buzz is a really deep scientist and consummate intellectual, as well as a great mentor. And so I signed up with Buzz, again, with the hope of turning out like that myself. And looking back, I can say it's not that easy to become like Buzz either. Now, the department itself was also amazing, really the center of the universe for many kinds of molecular biology at that time. The average person there was very impressive, and it was a tiny place, just 10 groups, and each group was very small. And so because of that, everyone basically learned about what everyone else was doing and learned and discussed and was involved. And I quickly grew to treasure that. And so I decided that when it came time for me to be a postdoc, I would want to be at another similar place with a wide range of expertise, very high specific activity, and where it was easy to learn and discuss with other people in these diverse fields. And at that time, aside from Stanford, the other place on the planet that was like that was the MRC lab in Cambridge, England. Now, I should say that, of course, whether to do a postdoc, that never occurred to me because, of course, one did a postdoc. That was just what you did on this escalator that led forward. So I decided that I would do a postdoc at the MRC, and that left only the question of with whom and on what topic. And then by chance, early or somewhere in the middle of my PhD career, Aaron Klug came through Stanford and presented a seminar. He had just discovered a novel structure and mechanics of some particular DNA sequence. And growing up, I'd met many beautiful and many really brilliant, beautifully brilliant people. But it was instantly clear that Aaron was up there with the very most brilliant of them. And I was really astonished by the depth and originality of his work and how it spanned the fields of molecular biology, chemistry, physics, and mathematics. And so it was obvious to me that I would like to be like that. And so even though by now I'd learned that proximity to deep people just isn't enough, nevertheless, I hoped that something would rub off and maybe I could be 1/100th or 1/1000th like that. And so, so I signed up with Aaron. Anyway, I'll spare you the details, but the short of it is I didn't manage to become like Aaron either, but I did learn a lot and have a wonderful time. And it was with Aaron at the MRC that I began my work in the area of chromatin structure, which has proven to be the single focus of our work over the past 25 years. And I'm going to stop there. And I just want to finally acknowledge my deep gratitude to you, my colleagues and troops, who for so many years have made it personally rewarding to wake up and go to school every day. Thank you.